To perform various qualitative tests on different amino acids and proteins, we will be performing a Ninhydrin test using Ninhydrin reagent, biurate test using copper sulfate and NaOH, xanthoprotic test using nitric acid and also NaOH, and the last one is sodium nitroprusside test using sodium nitroprusside and NaOH. This is to qualitatively assess the presence of different type of amino acids in the given substances. In addition to this, we will be needing various samples of amino acids, which you can see lined up here. In addition to this, we will be needing the test tubes, which we have labeled so that it becomes easy to identify, and we will be using a boiling water bath which will be placed in the fume cupboard. The first sample that we are going to take is glycine. So we will add 15 drops of glycine solution in the test tube. To this, we are going to add 5 drops of Ninhydrin solution. It's a 0.5% Ninhydrin solution in ethanol. We have added 5 drops of Ninhydrin solution. Now we are going to place this test tube in the water bath for 5 minutes. As you can see, the glycine solution has turned into blue purple color, which shows a positive test for Ninhydrin, indicating presence of alpha amino acids. We are going to perform this Ninhydrin test on another amino acid, that is proline. So we have taken a test tube which is labeled as proline, and we are now going to add 15 drops of proline solution in this test tube. We have added 15 drops of proline solution in this test tube, and now we are going to add 5 drops of Ninhydrin. We have added 5 drops of Ninhydrin to this proline solution, and now we are going to place this test tube in the water bath for 5 minutes and observe the color change. As you can see, for proline, the color obtained is yellow. It is specifically given by proline. So proline gives a yellow color at the end of this experiment. Now taken gelatin, and again we are going to add 15 drops of gelatin in this test tube, which is labeled as gelatin. We have taken about 15 drops of gelatin solution in this test tube, and now we are going to add 5 drops of Ninhydrin reagent. 5 drops of Ninhydrin reagent, and now we are going to place this test tube in the water bath. Unlike amino acids, you cannot see a purple color, even though there is a slight purple color, but that means it is very slowly developing because proteins are made up of amino acids. As you can see, different amino acids and proteins gave different type of tests. For the amino acids, we immediately got a dark purple color, as you can see, for tyrosine and glycine. For proline, because of its structure, different structure, it has got a yellow color which certains the presence of proline. As for the proteins, it also gave a purple color, but it took comparatively longer time to develop and it is little light in color.